Okay, in this session I'm going to be talking about how infectious agents get into cells, and in doing so they often alter the cell's normal cell cycle to benefit that particular infectious agent, and sometimes they enhance the cell cycle, sometimes they stop the cell cycle. So I'm going to be giving you some examples about how infectious agents can do one or the other. And this is summarised here. Now, in the previous session, we talked about PRB, cyclins, that's retinoblastoma protein, cyclins and cyclin-dependent kinases. And some viruses in particular might want to push a host cell into S phase so that that can aid in the viral genome replication. Whereas other agents, particularly quite a few intracellular bacteria, seem to want to stop the cell cycle and also prevent cell from undergoing programmed cell death or apoptosis, because that suits the functions of that particular infectious agent, to have the host not undergoing cell cycle progression. So here we've got human papillomavirus. You have to look at, if you're going to research this as part of your coursework, look at, does it have its own DNA polymerase, what sort of DNA replication machinery does the virus have, and what aspects of it does the host cell provide. And this is one example where the host cell is pushed into S phase because that really benefits replication of this DNA virus. Some intracellular bacteria, particularly I might mention chlamydia, inhibits cell cycle progression. These chlamydia develop into large intracellular vesicles, and the cell doesn't want to undergo cell cycle progression. The bacteria doesn't want the cell to undergo mitosis. That could disrupt the vesicle, and these bacteria take a couple of days to mature, and what the bacteria don't want is the cell to undergo cell division before that process has finished. So chlamydia, for example, will stop the cell cycle. So I'm going to take you through how one particular agent inactivates retinoblastoma, and if you're doing this as your essay topic, it is much more complicated than what I have described here. But the HPV, human papillomavirus, inactivates the retinoblastoma protein because it has a gene within its genome. So the viral genome is a circular genome, and these are all different genes that are coded from that. E6, E7, E2 are really important here. And the gene product of E7 directly inactivates PRB. In the last session you learned what PRB did, but PRB regulates cell cycle progression at the G1S boundary. If you remove all of the PRB from the cell, then all of the E2Fs are free to drive S phase progression, and it promotes early S phase. So by getting into the cell, producing the HPV E7 gene product, that protein will inactivate PRB and will get direct entry into S phase of the cell cycle. What we also find is the virus has got another gene called E2, and the E2 gene product is a negative regulator of E7. And what we find is when people get infected with this human papillomavirus, and especially the forms that cause cancer, is we actually get breaking of the genome here at the E2, within the E2 gene. So the E2 gene is disrupted, then this DNA spontaneously integrates into the genome of the host cell, and this is not part of the normal life cycle, this is just a genetic accident. We have the virus integrated into a cell, it's producing E7, it can't produce E2, and then the E7 has increased activity and completely abolishes PRB. So this is something that happens specifically in cervical cancer caused by human papillomavirus. And this is summarised here, and it links together the viral life cycle to the disease that it can cause. And here we've got normal epithelium, here we've got virus particles that get in, and in this colour here, sort of a purpley colour, is where we've got nuclei of cells, and we've got episomal viral DNA. That's DNA in the cytoplasm, that's how it should be, and it's part of the normal life cycle. Now what we find is that is what happens in a relatively benign lesion, also happens in a wart, because HPV causes warts, and that's what's happening here. Now what happens if you get integration of the viral genome into the host cell at the E2 site? Well, the E2 site is a negative regulator of E7, and if you get rid of, if you have excessive E7, you completely get rid of PRB. Now, it also causes overexpression of E6 as well. This loss of E2 results in overexpression of E6. That also takes out a protein called P53, which you'll learn about in a few weeks' time. Although if you're writing your essays on this topic, you'll probably want to research that now. But what we find is in disease caused by HPV, you get overexpression of E6 and E7 here. So we lose our PRB and P53 proteins that affect cell cycle in two different ways. Loss of PRB means you get cell cycle progression. Loss of P53 via this overexpression of E6 means that you can't induce P21, which is also another cell cycle regulator. So the end result is you get highly proliferative cells when you get infection with human papilloma virus. And when that forms a cancerous lesion, as shown here, then those two processes go way beyond what is normal for the normal viral life cycle. Here, the normal viral life cycle, you've got you've got E6 and E7 messing up P53 and PRB. Here, it's completely abolished. So I've mentioned that E6 disables P53. You might induce E6 might induce proliferation by P53 being unable to drive P21, as we heard in the last lecture. That is a cyclin-dependent kinase inhibitory protein. There's another protein called HPV-E5, and that drives epidermal growth factor receptor expression. So that will drive proliferation as well. And if you're going to study this particular virus, you might want to explore this activity or the E7 activity and look at exactly how that happens. But this slide just summarises the P53 aspect of it. So the HPV oncoprotein E6 inactivates P53, and without P53, your cells are less likely to undergo apoptosis, which is programmed cell death. They're less likely to stop the cell cycle because P53 induces P21. P21 takes out various cyclin-dependent kinases, and they're also less likely to repair DNA. So a cell passes through the G2M checkpoint despite the presence of mutations, and this is why the HPV virus can cause cancer, because cancer is a disease of accumulation of genetic mutations. So E2 is a natural negative regulator of E6 and E7, and if you get disruption of E2 by viral integration by complete accident, then you get overexpression of E6 and E7, and that's just like losing your PRB coding gene or your P53 coding gene. You get no protein. And in cancers, you tend to have gene inactivation of the PRB coding gene called RB1 and the TP53 gene that codes for this protein. What the virus is doing is almost replicating what happens in normal tumour formation. And this is an article out of Weinberg's The Biology of Cancer, just briefly outlining why certain viruses promote cell cycle progression. So pause the video and have a little read through that. So if you're going to if you're selected to do this particular topic as your coursework, you may want to be looking at some intracellular bacteria that mess up the cell cycle. And a good one to look into is Chlamydia trachomatis. Chlamydia is an interesting one. There's several ways in which it seems to be able to stop the cell cycle. And one of those is to infect cells and release a protease that can directly cleave cyclin B1. So as we've heard about in the last session, cyclin B is crucial for that metaphase to anaphase transition. So if cyclin B1 becomes cleaved by the Chlamydia trachomatis protease, then the cell will not undergo metaphase to anaphase transition, and the cell will remain stuck at the end of the G2M cycle of the cell cycle, which really benefits the chlamydia, because that needs 24 to 48 hours for those bacteria to form. If you research this, they go from a metabolically active reticulate body to a metabolically inactive but infectious elementary body, and that takes up to 48 or 72 hours. So it benefits the cell to be halting the cell cycle. So it has a few little tricks up its sleeve, and this is one of them. And this is another study, might be worth checking out this particular paper. And it talks about how the chlamydia trachomatis secrete a protease. It's a proteasome-like protease. We're going to cover the proteasome later in the semester, but this is a protein-degrading organelle, and the chlamydial proteasome called CPAF is thought to take out lots of proteins relating to cell cycle progression and survival. So if you are on the list to do this particular title, the infectious agents title, you have to cover at least one virus and at least one intracellular bacterium. Nice examples that you might want to look at would be herpes simplex virus. These are really interesting because you end up with multinucleated cells here, so there's clearly something going wrong with M phase, where the DNA seems to replicate. You end up with two separate nuclei, but then you don't get cytokinesis forming. So you might want to look into how the herpes virus does that. What's the molecular mechanism for messing up cytokinesis? Mycobacterium tuberculosis induces a long G1S arrest in persistent infections. So you might want to look into what proteins are secreted or produced by this particular bacterium to induce that G1S arrest. And P. gingivalis causes periodontal disease, and that also induces a G1S transition. So in effect, your task is to identify 3 interesting infectious agents, at least one virus, at least one intracellular bacterium. So it can be 2 bacteria or 2 viruses, or even, most likely that. And see how they affect either the cyclins, cyclin-dependent kinases, retinoblastoma protein, and outline the mechanism as to how it alters the cell cycle and why it alters cell cycle. It'd be really nice if you created a illustrated diagram of the signaling pathway and point out exactly where the infectious agent comes in and alters something. In the way that I've shown you of HPV, it's E6, E7 and E5. You can point out all of the points on that signaling pathway where that particular virus does it. But if you're going to do HPV, you've got to cover in great detail the proteins, some of the proteins and some of the accessory proteins that I've not talked about here. You might want to look up E6AP and some other related things. Okay, that's a summary of what I'm wanting for the coursework and how it links into the previous recording on cell cycle progression.